And especially in this context, because, sorry. At University of Chicago as well, I mean, the blood bank, transfusion medicine, it's intimately connected to immunology. I mean, a lot of the cells circulating, obviously, are immunologically relevant, and so it's a particular honor to be presenting in this forum. And just as a point of clarification, I didn't discover CTLA-4. We did work on it in the early days before, you know, most oncologists knew what CTLA-4 was. So I guess I'll take a little bit of credit for that. But yeah, so I want, and can you hear me okay? Right, is it projecting out there? Okay. What I want to present to sort of spin a story is really around bidirectional translational research and how cancer immunotherapy began, I would say, really as a bench-to-bedside enterprise, sort of making discoveries in basic immunology and then trying to figure out which of those, if any, might apply to the clinical context. And now it's flipped around to really be reverse translation, making critical observations out of patient material that generates new hypotheses that are worthy of testing back in the lab, which has given this slingshot effect where you can go very quickly then back from the lab back into the clinic with new therapeutic opportunities. So that's the flavor I want to try to give to this talk. These are disclosures. We do interact with a number of companies working on development of some of these immunotherapies. And just for the non-immunologists, I want to start with sort of what the underlying premise is with cancer immunotherapy. I mean, all of the successes that we know about are, or the vast majority, require CD8 positive T cells. So that's the aspect of cancer immunology and immunotherapy that we're focusing on. This is just a sort of a Google Images, version of a CD8 T cell engaging a tumor cell. This is an antigen-specific interaction. One of the, I think, important developments starting, it's more than 25 years ago, I think now, with Terry Boone and Steve Rosenberg identifying tumor antigens, was the molecular identification of sort of the recognition unit that CD8 T cells are using to see a tumor cell, delivering this kiss of death, as it were. It's important to realize and keep in mind that the antigens that are recognized by CD8 T cells are actually peptides derived from cellular proteins, most of them intracellular, presented by class 1 MHC molecules. For those peptides to be generated, they have to be processed from proteins. They have to traffic into the right compartment where newly synthesized class 1 passes through and gets to the surface, and there's a non-covalent association with beta-2 microglobulin. A lot has to happen to get that epitope, as it were, class 1 peptide complex to the surface, and it's important to keep that in mind when we think about mechanisms of resistance and especially secondary resistance. But the molecular identification of these antigens, again, more than 25 years ago, this generated the idea that why don't we just vaccinate patients, induce an immune response against those antigens, and then cancer will be taken care of, right? And so thousands of patients were put on vaccine clinical trials. We've had done some of that work, in the 90s, starting in the 90s. And, this is an example where we had metastatic melanoma patients. This is an easy image because it's cutaneous metastases for non-clinicians to see. You've got a bunch of lesions. This is a patient who had a complete response with one of these vaccines. These were shared antigens at the time, so these patients were immunized against a marker. It's called melin A that's expressed in melanocytes, but usually overexpressed in melanoma, but at least expressed in melanoma. In mouse models, these vaccines would induce complete responses in 100% of the mice, but obviously this is a rare event because after these 20 years, this is not an FDA-approved therapy. We had with our best vaccines about 10% of patients with bona fide clinical responses, about another 20% with 6 months stable disease or better, and everybody else progressed, right? So this is despite the fact that if you measured T cell responses in the blood, almost everybody had a strong T cell response. so there seemed to be a disconnect between the presence of circulating T cells capable of recognizing that tumor antigen, a tumor that had class 1 and the antigen expressed because they were pre-screened for that, and yet only a minority of patients responding. So this is sort of the backstory for thinking about the tumor microenvironment, where we reasoned then at the time, if you think even about like a two-compartment model of the tumor microenvironment and the draining lymph node, we as T-cell immunologists had always thought about lymph node, circulating T cells, right, priming and effector phase, but it was not a natural transition to think about the tumor microenvironment at that time, 20 years ago. And if you at least think about it as these two compartments, there's a growing tumor. If you vaccinate, it could be a vaccine draining lymph node, but we now know there are endogenous T cell responses against tumors. This wasn't well recognized at that time, that the initial priming of those T cells happens in a lymph node. We still think that that's the case, that most of the T cell initial priming happens in a lymph node. If those naive CD8 T cells or memory CD8 T cells see their antigen on an antigen-presenting cell, we now know more about the nature of those antigen-presenting cells. For the immunology aficionados, it's the Beta-3 lineage dendritic cells that are responsible for this cross-presentation. Those T cells then, they're activated, they differentiate, they expand, and then they leave into the blood circulation, and our immune monitoring assays were in that blood compartment, right? But the tumor's over here, and so for those T cells to execute the effector phase of that immune response, they at least have to traffic to the tumor. They have to find their way out of the vasculature, across the endothelium, into the tumor microenvironment, and we reasoned that at least chemokines, were going to be critical for that effector phase. But during this time is when some of the negative regulatory mechanisms were starting to be uncovered and better defined molecularly. And already we and others were finding that, and Liping Chen found B7H1, PD-L1 expressed in some tumors, reasoned that maybe those inhibitory mechanisms would have to be blocked too in order to sort of release the maximal function of those T cells once they did migrate in to translate into tumor regression. So the only way to start probing these aspects of the dynamic of the anti-tumor immune response system-wide was to biopsy tumors and study tumors. So this is a project that was actually hard to get NIH funded in the beginning, by the way. It's one of those things that maybe it's the most important experiment we did, was to actually biopsy tumors in immunotherapy trials. Hard to get NIH funded, but you keep pushing forward past that resistance sometimes. And what we found, just summarizing in this cartoon, is that, first of all, a large fraction of patients we found baseline already had an immune response primed against their tumor. And this was a subtlety that I think hadn't been so appreciated. Is that a fire alarm or something? Okay. Nobody else is getting up and moving to an exit, so I'm going to stay. And we know among those T cells that are, we call these the T cell inflamed tumor microenvironment phenotype. Among those T cells, we know are tumor antigen-specific T cells. I mean, since that time, obviously, this is the source of tumor-infiltrating lymphocyte-based adoptive therapy, where many patients have clinical responses. So in that pool of T cells, in the tumor, are T cells capable of recognizing and destroying the tumor? And yet, the tumor exists, right? These tumors are characterized by chemokines, some of which are recruiting those CD8 T cells, and a type 1 interferon signature that I'll come back to that's relevant to innate immune pathways that seem to be important for generating that spontaneous T cell response to begin with. And then, the philosophical question was, if there's been this, fairly vigorous immune response, why wasn't the tumor eliminated? And it's here that we found, again, from these initial gene expression profiling experiments and subsequent functional studies, that these are the tumors characterized by negative regulatory pathways that are upregulated. So the immune evasion for this category of tumor seems to be dominant inhibition, right? And in our vaccine trials, the handful of responders that we had had this phenotype to begin with, meaning the tumor microenvironment, once you vaccinated them could support recruitment of those effector cells into the tumor microenvironment. So it sort of wrapped up that and closed that mechanistic loop. Meaning also, if you could get just a few more T cells in, sometimes that could be enough to translate into regression. The non-T cell inflamed tumors lack all of those characteristics, and here our working model is actually, this is one of immune exclusion, where the immune escape mechanism is not letting the key elements of the immune system in. Those original studies were done with the first Affy gene chip technology that was available in our cancer center as a core facility, part of our genomics core facility at the time. But now, of course, with RNA-seq and TCGA, we have massive, and other public data sets, massive amounts of data available for different tumor types. This is the metastatic melanoma cohort among TCGA, and it's about 160 immune transcripts that are coordinately expressed that categorize this T-cell infiltrated subset, and it's about a third of melanoma metastases that have this indication of a strong immune infiltrate, which we've confirmed by immunohistochemistry and so on. And then it's sort of like a hot, medium, cold subset. There's subsetting. About a third of the patients have really, people call these cold tumors now, or no evidence of an endogenous immune response. We call these like the rocks, where it's like hard to squeeze a T cell into those tumors. And I'll show you some sort of mechanistic data arguing for that resistance. So this is the subset that we think stands to benefit most from interfering with negative regulation, and those kinds of data come from that kind of idea comes from these sorts of data, which is that if you then start probing these candidate negative regulatory pathways by immunohistochemistry, or now fancier multispectral immune fluorescence in tissues. When you have CD8 T cells, sometimes patchy in a patchy fashion around the periphery, but in some of our melanoma metastases, they're like CD8 T cells throughout the metastasis. Even if it's patchy, it's in those regions where there are CD8 T cells that you see FOXP3 regulatory T cells, so the suppressor T cells that can turn those effector cells, or at least back off, or at least limit their function. PD-L1 is an inhibitory receptor, ligating the inhibitory receptor PD-1 in melanoma, sort of diffuse on T cells and on tumor cells and non-tumor cells, myeloid cells, and then IDO, indolamine 2,3 dioxygenase. This is a tryptophan catabolizing enzyme that also has been linked to peripheral tolerance, also staining here mostly myeloid cells in the same tumor microenvironment. It took a while to characterize enough tumors and come around to the view that the non-T cell inflamed tumors generally in melanoma lack these factors, right? And so as we had sort of embarked on this initial work from the hypothesis perspective, the general feeling was that the tumor must set up an immune suppressive tumor microenvironment, and sort of like a catcher's mitt, waiting for the T cells to come in, and that way whenever a T cell came in, they would be turned off, right? But these data suggest that this is not a tumor-driven process, because the CD8 T cells have to get there first. So it's really an immune intrinsic negative feedback loop. And we had mapped in mechanistic studies that it's gamma interferon made by the CD8 T cells that are upregulating PD-L1 and IDO is sort of a functional unit, and chemokines, including CCL22, recruiting most of the regulatory T cells into the tumor microenvironment. And so without CD8 T cells, these are not upregulated in the tumors. And these kinds of data during this time were suggesting, well, maybe then it's possible to, because we know there are tumor antigen-specific T cells in those tumors, and if they're held in check by negative regulation, maybe we can block that negative regulation and regain some functional attributes. The first to get focused upon was PD-1 interactions. Mechanistically, we've done, and others obviously as well, Sasha Rudensky, getting rid of FOXP3 cells in mouse models, and I'll show you some data in IDO, and then there are others. So demonstrating mechanistically in preclinical models, these could all be sufficient to get improved anti-tumor immunity. And In the early days, when the pathway was first defined and we didn't have antibodies available yet, like Li Ping's B7H1 antibody, there was a knockout mouse that had been made by Hanjo's lab in Japan, and we were able to get those knockout mice sort of before people were interested in anti-tumor effects. And in some of our models, just getting rid of PD-1 on specific T cells could result in complete spontaneous tumor elimination, even under situations where CTLA-4 deficiency had much less of a therapeutic effect. And so in these early days, it was hard to get people interested in trying to block PD-1 as a therapeutic, and now it's hard to find somebody who's not interested in blocking PD-1 as a therapeutic. And it's sort of fun. We were part of that early day. And just to get all the non-clinicians on the same page, obviously this has been, maybe even transformational in cancer therapies. The first FDA approval in melanoma in 2014. Now we have FDA approvals in 9 additional cancer entities and counting with just single-agent anti-PD-1. So broad applicability to multiple cancer types. I would say in part because this biology is sort of hardwired into the way that the immune system works. These are immune intrinsic regulatory loops, not sort of cancer cell driven. All right. So with that as sort of the background then, one, we can think about how good is this T-cell inflamed gene signature as a predictive biomarker. So Merck has imported this concept into with the nanostring platform, which has pretty good performance with paraffin tissue, and these are beyond melanoma now, head and neck cancer, gastric cancer. Tanguy Seiwert at our place was active in these studies, and you can see if patients' baseline tumor biopsy is low for this T-cell interferon gene signature, almost none of those patients go on to respond to anti-PD-1 single agent. And the responders are enriched in those who have this T-cell inflamed tumor microenvironment gene signature at baseline. So that's true in gastric cancer and several other cancers, including melanoma. So now you can think about if we're going to make anti-PD-1 better, how can we improve upon this, right? And so the way we, in sort of a simple-minded way, divide this up is we need to find a way to capture more of the T-cell infiltrated patients, right, to gain a larger fraction of response, or maybe, to get all of them to respond with the T-cell inflamed subset. And then we may need very new ways of thinking about the non-T-cell inflamed or the cold tumors biologically to try and gain any kind of clinical activity in that subset. And I'll dispense with one concept here. We had thought for a while that maybe the cold tumors or the non-T-cell inflamed tumors lack antigens. Maybe they just have no mutational antigens or differentiation antigens or what have you. We've studied that quite a bit and have published one detailed paper about this, but we don't think that the non-T-cell inflamed tumors have a shortage of antigen. So it's not lack of antigen. Which is good for us. That gives us a lot of opportunity to try and get immunotherapy sort of cooking in the non-T-cell inflamed subset. So in terms of the T-cell inflamed subset then, how can we do better than anti-PD-1? So we had been attracted to the idea of combinatorial blockade of PD-1 and IDO since the original observations that they're coordinately expressed, and this had to await a drug, and we were fortunate enough to establish a collaboration with a company, Incyte Pharmaceuticals, who were the first to make an orally bioavailable potent IDO1 inhibitor that in mice was capable of blocking the pathway system-wide, right? So we worked with them, got the IDO inhibitor for preclinical experiments, and I'm just showing a couple of highlights of data. These are pre-established B16 melanoma models in mice. where, in the not too long ago, it was hard to get anything to give any, tumor control in established B16 melanoma. But here now, if we combine single agent, whoops, it's kind of spastic here, the mouse, IDO inhibitor with anti-CTLA-4 antibody or with anti-PD-L1, we now have additive or synergistic effects where some of these mice have complete tumor elimination that, those mice are then protected against subsequent rechallenge. So both of these combinations have gone into patients. We did a phase 1 trial along with UPenn with single agent IDO inhibitor, dosed it until we knew we were hitting the pathway, because IDO is a tryptophan catabolizing enzyme, and one of the metabolic products is kynurenine. So you could measure the flip in tryptophan to kynurenine levels in humans, right? And you could see that flipped, so we knew there were biologically active doses. Both of these antibody then combinations went into phase 1 and phase 2 expansions, and in both cases the response rate is almost double what you would expect with the antibody alone with the single agent or the single arm trials with pembrolizumab combination. These are the early data shown here, but they've held up with as the cohort matured. The response rate is around 56% in metastatic melanoma, IDO inhibitor plus anti-PD-1. You can see now some of these patients at their first CAT scan already are having CRs, right, that seem durable. So this is an example of a combination. This has gone to a phase 3 trial. The melanoma phase 3 trial is accrued. We'll see, that's going to be the proof, whether the combination is better than single agent, but several other phase 3s are also in other cancers are in the works, and several other companies now have IDO programs. We're participating in the BMS IDO inhibitor anti-PD-1 nivolumab trial, which also looks interesting so far. So that's one example of combinations. As we've probed in on the T cells, I'm not going to spend a lot of time on this. The basic immunologist, some of us, we've talked about it sort of offline during my visit here, but we realized that there's speculation around, the T cells in the tumor microenvironment, they just must be completely inert, right? Functionally incapable of doing anything. We've found that that's not true at all. A lot of that inference has come from chronic viral infection models, like the chronic LCMV model in mice, where the T cell exhaustion phenotype has been defined, and those T cells can't lyse, they can't make interferon, they can't proliferate. They're probably terminally differentiated cells targeted for elimination. We found that the dysfunctional T cells in the tumor microenvironment retain a lot of these properties. They make gamma interferon, and we know that's the gamma interferon that's inducing the PD-L1 and IDO. They constitutively are making chemokines that are recruiting Tregs. So we've shown this now by sorting those TIL. Anyhow, so the reason for the failed tumor rejection is not because they're functionally inert. They're probably actually contributing to immune regulation in the tumor microenvironment. But by identifying markers, we could sort the tumor antigen-specific TIL from the tumor microenvironment and did RNA-seq and compared them to other data sets, and now have a collection. It's over 100 surface proteins, genes encoding cell surface proteins that you can use to characterize those cells. And some of these are known. It turns out when we sort them out, in contrast to the chronic LCMV model, I'm just summarizing here, these cells do have an array of inhibitory receptors on their surface, so PD-1 and CTLA-4. Those of you in the field probably recognize the names of some of the other defined ones that also are in the clinic in terms of antibody targeting. LAG3, TIM3, I know Vijay Kutru was here yesterday, maybe he talked about TIM3, I'm not sure. TIGIT 2B4, which in this context is probably inhibitory. But unexpectedly, these T cells also have a whole series of co-stimulatory receptors displayed on their surface, which is different from the chronic viral infection model. And several of these, all of these have antibodies in the clinic now trying to push on them as co-stimulatory receptors And I should add here that even with the costimulatory ligand agonistic antibodies as an immunotherapy, we have shown in preclinical models that the therapeutic effect is all driven by T cells already in the tumor microenvironment. You can have additional priming and draining lymph nodes, but you can get rid of those T cells or keep them in the lymph node with a poison like FTY720 so they don't traffic out. And all the T cells that are in the tumor already are sufficient to do the job. So even the agonistic antibodies that you think are helping to prime new T cells, for this context, it's sufficient for them to tickle the receptor on the T cells that are already in the tumor microenvironment. And then through this analysis, we got a bunch of new potential targets that are fun projects now sifting through, and some of them have really different kinds of biologic properties. So all that to say that we'll have, I think when the dust settles here, we'll have multiple combinations trying to touch these, reactivate or restore the optimal function of the tumor-infiltrating CD8s in the T-cell inflamed subset. And just to show you don't have to touch a PD-1 or CTLA-4 to have a therapeutic effect. Preclinically, if you take two of these markers, blocking antibody against LAG3, agonistic antibody against 4-1BB, you can again flatline these B16 melanoma tumors and other tumor models. So some of these combinations could be interesting later when we're trying to rescue patients who are refractory to anti-PD-1 single agents. Maybe some of these other pathways are going to be functionally relevant. And again, all of this still succeeds if you block new T cell egress from lymph nodes, and can all be efficacious just on T cells in the tumor microenvironment. So that's the optimism now, around the T cell inflamed subset. So the rest of the talk, and really I think the harder part of the field, the bigger challenge is the non-T cell inflamed subset. And so how can we wake up or initiate or catalyze an immune response, uncouple the blocks on the immune response in non-T cell inflamed tumors to get this endogenous immune response going, which still might be held in check by negative regulation, but we know how to block that now, right? And so how do we jumpstart this immune response? So one approach that we've taken is to think about, zoom out and pause for a second. How is it possible that there ever could be a spontaneous T cell response against the tumor because there's no pathogen? So for an adaptive T cell response to be generated, it's thought that you need an innate immune recognition to trigger activation of dendritic cells so that they could properly co-stimulate T cells and get T cells going. So we wondered, what could that pathway be that was driving an endogenous T cell response against a tumor? And so we took as a hint the observation that in our original gene expression profiling of tumors, the CD8 infiltrated tumors also had evidence for type 1 interferon gene signature. And type 1 interferons are they're secreted, transcribed, genes are transcribed downstream from innate immune pathways in antigen-presenting cells. So we wondered, could absence of a spontaneous T cell response be because innate immune pathways are not being engaged, and what's the innate immune pathway? So I'm just going to summarize a bit of the mechanistic data, which took a couple of years to resolve. And what we found basically was that those type 1 interferons themselves, interferon beta in the mouse models, were necessary for spontaneous T cell priming against tumors. So that type 1 interferon signature, that's a requisite part of making a spontaneous CD8 T cell response against a tumor. All right. And then once we knew that, we basically could go through candidate upstream innate immune pathways, TLR pathways, TLR4, TLR9, MyD88, TRIF adapters, RNA, cytosolic RNA sensing, right, through the RIG-I pathway. There was a model that extracellular ATP spilled from cells might activate innate immune pathways. We were able to line up these knockout mice, put in a tumor, measure type 1 interferons, and measure our T cell response, and we found no defect in any of those mice. And so that led us to what at the time was a recently described pathway, which is the STING pathway. And the STING pathway is part of a cytosolic DNA sensing pathway, and had been identified as important for recognition of DNA viruses to make immune responses against DNA viruses. And sting knockout mice are highly susceptible to DNA viruses. The main DNA sensor is probably C-gas, and C-gas generates cyclic dinucleotides, CGAMP, which then triggers sting aggregation, and then there's the rest of this cascade that leads to type 1 interferon production. So when we put tumors in sting knockout mice or IRF3 knockout mice, we lost the T cell response, right? And so this seemed to be one of the more important pathways for innate immune sensing of tumors. We found that we could identify labeled tumor-derived DNA in the cytosol of tumor-infiltrating dendritic cells. About 30% of dendritic cells in the tumor have tumor-derived nuclear DNA in their cytosol. We don't know for sure that it's DNA transfer. It could be that cyclic dinucleotides themselves get transferred between cells. So we haven't proven that DNA is the ligand derived from the tumor. It could be CGAMP that's derived from the tumor. So we're still working on that detail. But this whole model then, if you'll allow me the model, then generated the idea that maybe we could, if we could develop agonists to push on the sting pathway, maybe that could be a cancer therapeutic. Maybe that could wake up the cold tumor microenvironment. So here too, this is a value of going to meetings and going to poster sessions and talking to people in the corridors, right? We almost, serendipitously hooked up with a company, Aduro Biotech, who it turns out they were working on sting agonists as a vaccine adjuvant, like to mix with antigen to immunize, right? And then we showed them our data and realized maybe they have a cancer drug. And so starting with a tool compound, which we have now shown is a sting agonist, we could set up these pre-established tumor models. This is B16 melanoma again. We've done this with 5 different transplantable tumor models, and you can see now in these experiments, this is just a single injection of a sting agonist into the tumor, and you can see all of these mice have their tumor completely eliminated, right? And all of that depends on host sting. So this is tumor growth in sting knockout mice. So it's not sting in the tumor cells in these models, it's sting in the host. And it's in the host antigen presenting cells largely. This is the augmentation of the endogenous immune response. So this is the endogenous immune response we're normally working with, just defining the T cell inflamed, hosts. Here it's, logs increase in both, and this is in the tumor, beyond that endogenous immune response. All of that is ablated in sting knockout mice. So we worked with Aduro then to help screen compounds against human cells, human antigen presenting cells. It turns out the human sting gene is polymorphic, and so we were able to genotype donors, and this is normal donors, and then be sure that the compound selected could engage all of the polymorphic variants of human sting. Tested that in the mouse models. That's all part of this paper from 2015. And so now that Aduro drug, now with the backing of Novartis, is the first sting agonist in phase 1 in patients. It's being given intratumorally. And already, as the dose levels are increasing, it's being combined with anti-PD-1. With the thinking that if you push a new immune response, especially with the gamma interferon plus the type 1 interferons, almost for sure these same negative regulators are going to come in and circle back. And so the anti-PD-1 combination was sort of engineered into the protocol from the beginning. And now the other groups have sting agonists now that are in the clinic also being combined with anti-PD-1, including Merck. And so we don't know yet, how this is going to play out. For us, it's a very strong candidate for an immune activator. Some of you know we've worked with T-VEC, the oncolytic virus, injected into the tumor combined with anti-PD-1. That's engaging the sting pathway almost for sure as a herpes virus. And other TLR agonists are being squirted in tumors now, all zombie drugs that are being rejuvenated now to manipulate the tumor microenvironment now to try and combine with anti-PD-1. So that's one strategy. Maybe we can promote a de novo innate immune response in the tumor, get the immune response going, and then block checkpoints and gain efficacy. So stay tuned as that progresses.
Tom, when you do those experiments, typically selecting people in advance with cold tumors? You know they're cold? Or?
No, so the question was, are we pre-selecting patients at all? We're not selecting patients at all, based on a cold tumor phenotype. Nobody knows to what extent they might be active in which subsets of patients. When you design these phase 1 trials, especially with an injectable drug, you have to lower the activation energy, as much as you can. So it's patients with injectable tumors. In the beginning, it's all visible cutaneous or lymph node masses, recurrent primary breast tumors, Merkel cell patients, melanoma patients, some lymphoma patients. So it's a diverse spectrum, but the primary endpoints here are biopsy driven, right? We want to do pre-biopsies, on-treatment biopsies, see if we started off with a cold tumor, does it now have, how far did we get in this cascade? Do we have type 1 interferon gene expression? Do we have T cells coming in, right, and so on? So it's sort of first principles driven. So then the rest of the way we've approached this idea of the cold tumors is to go back to the patient population. And so if you imagine, like these, I see melanoma patients, these are 2 of my patients, these are lung metastases, right? One patient, this is CD8 immunohistochemistry, full of T cells all throughout their tumor, another patient, none. Why might those 2 patients be different, right? And, because this patient is likely to go on and be a responder. So it could be that the tumor cells are different, so somatic variants. In cancer, we have a lot of reasons, to have somatic differences between tumors, and in particular, we were interested in the idea of maybe there were oncogene pathways which might be on in tumors that were keeping the immune response out, right? So that's one. Germline differences, so there are germline polymorphisms linked to autoimmunity, so why not spontaneous anti-tumor immunity, which in many respects is a little bit like autoimmunity. So this is something that could be, assessed experimentally. And then environmental differences. And in the beginning, when we set up this schema, this was sort of a blank, spot. We didn't have a good candidates environmental factor until some of the emerging work from Dan Littman and others, the Favell lab here, about the role of the commensal microbiota in systemic immune responses that made us realize, well, maybe commensal microbiota could impact on global anti-tumor immunity. And so all of these are tractable. We've expanded our own in-house biobanking. Everybody who's going on an immunotherapy, as many of those patients as possible, we have an infrastructure to consent them and bank all of the tissues, including serum and peripheral blood cells, but including stool now for microbiome sequencing. When we started our immune monitoring core, 20 years ago, I had never thought that we would now have a stool room, where, and the stool samples, they take up more space, each sample. So you need more freezers for stool than you need for, blood samples or tumor biopsies. So whole operation now for microbiome analysis. So this is prospectively being done in as many patients as possible with as many cancers as possible. But in the beginning, before we had enough of our in-house samples, we realized that some of these questions could be addressed in part using public tumor sequence databases like TCGA. And so TCGA is super valuable because across, now it's well over 10,000 tumors of most histologies, not all histologies, but you have RNA-seq data on those tumors, which means we could score them based on our immune gene signature, right? So our phenotype is the degree of this spontaneous immune response. So that we can categorize, these are the melanoma patients in TCGA, so metastatic melanoma, and categorize them as sort of hot, medium, cold, and analyze the non-T cell inflamed tumors now as a phenotype, and now look at genotype, right? So at the tumor level, we could look at the exome sequencing data and see if there were any oncogene pathways disproportionately represented in the cold tumors, right? At the germline level, we could look at germline SNPs the same way, right? Because those data are also in TCGA. So public data. When we did this with our first melanoma study, what we found was that we could find a mechanism, candidate mechanism for Wnt beta-catenin activation in almost half of the cold melanomas, the non-T-cell inflamed melanomas. And the lead there came from the small number of tumors that had activating mutations in beta-catenin itself. And that's only 8% of the tumors, but it was enriched only in the cold tumors, right? So that led to a candidate analysis to look at beta-catenin in the rest, and as well as a beta-catenin target gene signature. So that's a strong correlation, but how could we prove whether that was causal for T-cell exclusion, right? And it could just be an epiphenomenon. So to examine this question, we turned to genetically engineered mice, the active BRAF P10 knockout mice that was originally generated by Marcus Bosenberg and colleagues. And when we engineered a model antigen into those mice, so we had Cre-inducible expression of an antigen as well, we found that indeed there could be spontaneous T-cell priming in that basic model. And I briefly mentioned beta-3 dendritic cells. We found that this was dependent on beta-3 dendritic cells in those mice. We could do that by bone marrow chimeras with beta-3 knockout bone marrow. And in our hands in these mice, those T cells could be sufficient to get some therapeutic activity of tumor shrinkage, not with single agent checkpoints, but with combination PD1, CTLA4, dual blockade, we could get tumor shrinkage. So these were responsive to checkpoint blockade, just like a patient might be responsive, although maybe patients are even more responsive. If the only difference was the mice were engineered so that active beta-catenin was on in the tumors, so the rest is the same, none of this happens. There's no T-cell priming. The reason there's no T-cell priming is because there are no beta-3 dendritic cells recruited into the tumor microenvironment. These tumors, there's no T-cell infiltrate at baseline. You treat with checkpoints. There's no tumor shrinkage, and there are no T-cells that come in after treatment. Right? So this could very well be a resistant patient, right? And we worked out some of the detailed mechanism about how at least one way beta-catenin signaling is keeping the immune cells out. All right. So this is our best cold tumor model. All right, where it's hard to get anything to work in that model. So it's a useful screen, if you will, or a testing ground for new interventions. Now, one thing that came up was whether, there's no spontaneous immune response made in those mice, and people are saying, well, clinically in the cold tumor phenotype, maybe they haven't made a T-cell response at all, so all you have to do is vaccinate them, like with the neoepitope vaccine, and then maybe you'll gain responders. Or maybe you could grow up some T-cells and adoptively transfer them and then gain responders, right? So we realized we could model that or address that kind of question in this model. And so we have the mice that are engineered to have this model antigen, SIY, and when we transfer specific T cells, we can get therapeutic effect of the beta-catenin negative tumors. But this completely fails if the only difference is that beta-catenin is on in the tumor cells. And same thing with vaccination. We vaccinated these mice prophylactically or therapeutically. Nothing happens in the beta-catenin tumors, and the reason is the effector T cells never go into the tumor. So remember this issue of T cell migration into the tumor microenvironment. The best indicator of this is maybe on the right, where we've normalized per gram of tumor, because some of the tumors are shrinking and some aren't, so this is a good way to normalize data in the tumor microenvironment. And the number of transferred T cells is very close to zero in the beta-catenin expressing tumors. Why is that? Why do effector T cells not go into those beta-catenin tumors? Well, it turns out because of the same reason. It's the beta-3 dendritic cells. We found in this series of studies, when we sorted the different cells from the tumor microenvironment, the main factory making the chemokines to recruit effector T cells was the beta-3 dendritic cells. We could rescue everything if we just inject one dose of activated beta-3 dendritic cells into the tumor. Everything follows. The T cells are primed, the effector T cells go in, checkpoint blockade works, and everything. So this is the key rate-limiting step, we think. Right? The non-T-cell inflamed human tumors also seem to lack beta-3 dendritic cells, and we've started working this out as one of our immunofluorescent panels, staining tumors. And it's still a work in progress, but we've found a BATF3 antibody that works well by immunohistochemistry. You can see nuclear staining in a subset of cells, and you see in some of these samples, beautiful, this is a bladder cancer patient, beautiful clusters of CD8 T cells and nuclear BATF3 staining cells in real tumors in patients. And this particular patient went on to be a responder to checkpoint antibodies. We're studying a little bit more the function of those Beta-3 DCs in the tumor microenvironment beyond chemokine production for T-cell migration, and we think they have additional functions as well. So this beta-catenin inverse correlation goes beyond melanoma. This is a study along with one of my colleagues, Jason Luke, where we've gone through all the tumors in TCGA, scored them for presence or absence of the T-cell gene signature, and in at least 22 of these tumors, there's an inverse correlation between beta-catenin pathway activation and T-cell infiltration. It turns out one of the easiest way to gauge that, it turns out part of TCGA was reverse phase protein arrays, and this was largely done to screen for tumor suppressor genes that might be absent at the protein level, like P10. It turns out beta-catenin was on that list, and so there is beta-catenin, like whole tumor lysate data on 10,000 tumors out there in the cloud, right? And it turns out just beta-catenin protein expression inversely correlates with, not transcripts, but protein with the T-cell infiltrate. And Stephanie Spranger, who has finished in my lab and started her own group at MIT now, and I recently went through the literature this is now a subfield of tumor immunology, is immune-evasive oncogene pathways, and summarized the pretty solid data on these additional pathways that are contributing to immune evasion. All right, I'm going to skip this for now. I'm using too many words today, and give you one slide on SNPs. This is work in progress. This is where we have the least data, but have one interesting lead. Again, in melanoma, just using TCGA, we were able to find a polymorphism that crossed sort of genome-wide significance. This is in a gene that's been associated with autoimmunity, which was sort of the hypothesis. We have a knockout mouse model. We've generated bone marrow chimeras, so we have that gene deleted only in hematopoietic cells. And just with that as a single intervention, there's better spontaneous immune control of tumors already. It turns out this is a druggable target, so it's raising the idea that maybe there are germline encoded variants that we can sort of phenocopy with drugs and also turn into therapies. So that's just a little bit of a teaser, a snippet, but to prompt maybe positively reinforce banking germline DNA from patients. So I want to spend just 5 minutes on the microbiota, if that's okay, where the idea here was, could there be this environmental difference also contributing to spontaneous immune responses, or the degree of that happening? And the rationale, again, is that commensal microbiota composition had already been linked to autoimmune diseases in various compartments beyond the gut. So it's not just colitis, but autoimmunity in other sites. So might this be relevant for anti-tumor immunity? So in mouse experiments, we basically started with buying essentially genetically identical mice from 2 different vendors that had already been published by Dan Lippman to have different microbial compositions, right, among the commensals. And when we did that, these are mice from, those of you who do mouse experiments, from Jackson Labs or Taconic. And what we found was that, this is B16 melanoma again, tumors always grew slightly faster, but significantly in mice from Taconic, right? And this was associated, so this slightly slower tumor growth in Jackson mice was associated with about a 2- or 3-fold increase in the endogenous T-cell response against the tumor. So not shown here, but that was enough to translate into a huge difference of anti-PD-1 efficacy. In Jackson mice, anti-PD-1 single agent, complete responses, right? We had always used Taconic mice because they were cheaper, and Taconic mice single agent gives you a little bit of a tumor shrinkage, but never complete responses. And all of this difference is microbiome mediated. You could eliminate it by co-housing for 2 weeks. The Taconic mice got better, So the mice with the inferior microbiota, it was a gain of function when they were cohabitating with Jackson mice. You could also transfer improved efficacy by fecal transfer from Jackson mice into Taconic. This is pre-transfer, so before we put the tumor in, we do a fecal transfer, and there's already better spontaneous control in Taconic mice, and the reverse transfer does very little. So it's mostly a gain of function if you have good microbes. We did sequencing and some different, in different permutations. There are about a dozen bacteria that were enriched in the mice that were stably recolonized with new bacteria from Jackson mice, and one of these was Bifidobacterium, and so this is something we can get our hands on, different strains of Bifidobacterium, and indeed we found now with pre-established tumors, these are Taconic mice, where, as I mentioned, anti-PD-1 or PD-L1 gives very little marginal effect on its own. Bifidobacterium alone with a pre-established tumor now also gives some therapeutic effect, and then you combine them together, and now you regain the near CR rate that you get in Jackson mice, right? And so all of this is immune mediated. It's not a direct anti-tumor effect of the bacteria. If you get rid of T cells, everything is that benefit is all eliminated. And we're not saying Bifidobacterium is the only bacteria. There are multiple that we're correlating. It's actually a work in progress, screening some of the other bacteria. But this gave us the opportunity to check this in patients. And I should add that most of the data in these mice support a model whereby the favorable gut microbiota are resulting in slight pre-activation of dendritic cells in the entire animal that appears over the course of days to a week after bacteria transfer by gavage, right? And that leads to this slight increase in the spontaneous T cell response, which gives you more substrate on which anti-PD-1 or PD-L1 can act. So that's the working model that we have as we're funneling through mechanisms. So we just completed the first cohort of human patients. These are metastatic melanoma patients. It's a small cohort. We started analyzing when we hit 50 patients just to start to see what we had. And we were surprised, I was surprised at least, to find that we already had, by 16S sequencing of those baseline stool samples, bacteria that were enriched in responders or enriched in non-responders. It turns out one of those was a Bifidobacterium OTU. That's the top hit here by coincidence. We did metagenomic shotgun sequencing and then developed species-specific PCR primers to try try 3 different assays to confirm some of these bacteria. The Bifidobacterium subtype appears to be, by correlation, Bifidobacterium longum. So we worked out species-specific primers and found that the PCR also correlated. So Bifidobacterium longum, at least as a species, is probably the one that's correlating. We did this with 10 total species where we confirmed by species-specific PCR, so all 3 assays are aligned. And it turns out if you use that as a biomarker now, so this is baseline stool sample analysis, right? And then patients go on to get anti-PD-1, and then you score if they're a responder or non-responder by RECIST. And it turns out if we just take these 10 bacterial PCR reactions now as a multiplex, that splits out responders and non-responders quite well. And in these, that's for the whole cohort. In 15 patients, we had baseline tumor biopsies that passed quality control, and so we could look at mutation load, at trends in favor of responder, PD-L1 expression, PD-1 expression, CD8 quantitative immunohistochemistry. These all trend in favor of responder, but statistically, these different reflections of the microbiota is actually the best of the predictive biomarkers in this cohort. So not what we expected at all. Now we're thinking maybe the microbiome is more of a major push for the endogenous immune response to favor anti-PD-1 efficacy. And then maybe the most surprising kind of result is now the mechanistic aspect. Like, how do you know if that's just an epiphenomenon or causal? So the way we've been doing this is by transferring human fecal material into germ-free mice that don't have their own microbiota. And if you do that, we have several examples of this now. If the patient went on to be a non-responder, that fecal material and microbiota transferred into germ-free mice. You let it equilibrate for a couple weeks, put in a mouse tumor, and treat with mouse anti-PD-L1. That mouse is a non-responder, right? If the mouse, the germ-free mice, got fecal material from a patient who went on to become a responder, there's a higher spontaneous immune response. There's slightly better, spontaneous tumor control, and that mouse is a PD-L1 responder. Almost as if the microbiome composition is a gatekeeper for that immune response, meaning that maybe it's something that we could manipulate. And so we're working with another startup company, Evelo, to try and identify bacteria that could be developed as kind of a probiotic, but not in the traditional sense of a probiotic, a bacterial-based intervention to try and improve endogenous immune responses to then gain better efficacy. So what I tried to argue was that just thinking about the T-cell inflamed tumor microenvironment has, I think, led us down these different avenues that they've definitely been fun basic immunology projects, but also I think with relevance for new interventions. The farthest along is IDO, is target to co-inhibit with PD-1 blockade. By studying those T cells in more detail, I didn't show you a lot of any of the primary data, but there are multiple other regulatory proteins that we can consider targeting as well for the T cell inflamed. For the non-T cell inflamed, we have STING agonists and other innate immune activators that are in the clinic now to try and push those tumors. But maybe we can also drug oncogene pathways that are immune exclusionary, right? To be more surgical or precise about how we're uncoupling these escape pathways in the tumor. So, and then the multidimensional analysis of patient material is pointing us to kind of a comprehensive look. Our idea is that maybe we can find sort of individualized resistance mechanisms in each patient, and then you kind of assign the right drug and maybe ultimately have the perfect combination for each patient, including the microbiota. So I think I've been talking a long time. I'll leave the acknowledgments up. I mentioned Stephanie Spranger, who did the GEM models, really was very productive postdoc in the lab. The microbiome stuff that I just mentioned, Viara Matson, Jessica Fessler, postdoc and a graduate student, both working hard now on the mechanism and the specific bacteria. The older work about the STING pathway, Leticia Corrales and Seung-Yong Wu, who have both gone on from the lab at this point. So thanks for your attention, and if there's any time, I'll take some questions. Thanks.